All right, welcome everyone to this week's Wednesday webinar. We offer on the second and fourth Wednesday of each month. This is part of the Mid-Atlantic Women in Agriculture Network. And we'd like to give a shout out to our sponsors of the program, as well as there's a number of regional collaborators. And so a big thank you to them for their support of this project. Today, we have Amanda Grev that'll be speaking on forage sampling and forage analysis. You can access the upcoming webinars and see what we're offering through the December, or you can go to the blue link here and you can see all of our archive webinars. So they're all posted on our YouTube channel. You can check out the last, I think we've got 4 or 5 years now of webinars cataloged, and then again, we've got them planned through December. So with that, I'm going to turn this week's presentation over to Dr. Amanda Grev. We're really happy to have her here today and to talk about forage, and a very important part of our livestock's diet. So.
All right, thanks, Jen. Does it look okay?
Yes, looks great.
Okay, perfect. So welcome everybody. I see from the chat that we have, a few people joining from overseas too, which is cool to see. So thank you to everyone for joining us. And like Shannon said, today we're going to talk about forage sampling and forage analysis interpretation. So really quick, kind of before we get into kind of the how-tos and the, actual analysis, I wanted to talk briefly about kind of forage quality and some of the terms associated with it. We always, or oftentimes we use, terms like forage nutritive value, forage quality, kind of interchangeably. But actually there is, somewhat of a difference between these different terms. So, forage nutritive value is kind of like the actual concentration of nutrients. So, how much energy, how much protein, how much fiber, how many vitamins and minerals are in a forage. And forage quality is kind of a broader term that includes that nutritive value, but also, a lot of other factors that we'll kind of get into in a minute. So I like to think of it as, forage nutritive value is kind of like the nutrition label, that you see on a lot of our human foods. It tells us exactly what ingredients and what nutrients are present in that food. So forage quality can be influenced by, a number of different factors. We have things like palatability. If we have a really highly nutritious forage, but the animals aren't going to eat it, that's not really going to do us a lot of good, right? So that's a factor. We have intake. How much will they eat? How much of that forage can they consume before, gut fill and other things kind of slow them down? And digestibility. How much of that forage is actually going to be digested and broken down and those nutrients be made available to the animal? And of course, the nutritive value, which we just kind of said was like that, nutrition label, the actual amount of nutrients, is that adequate for our animals? Are we providing enough nutrients that way? And then there's also things like anti-quality factors, which are, compounds that may cause issues, whether those are different toxicities, weeds that are present, other debris that may be kind of, made it into the hay or something through the haymaking process. Those are all kind of things to consider when we're talking about, forage quality as a whole. So I like to think about forage quality as, the extent to which a forage has the potential to produce the desired animal response. And that desired response may be slightly different depending on what type of species we have. But if we're getting to where we want to be going, then we're doing well in terms of forage quality. So for those of you who might be a little bit more visual, if we look at this from kind of a visual perspective, like I said, there are a whole lot of things that are going into that end goal of that animal performance that you see at that top of that figure. We have a whole host of all of these different factors. The forage nutritive value is one of those many things. And the forage quality is kind of that larger picture, all those factors coming together and resulting in that end unit or that end animal performance. So really, when we think about forage quality, we kind of have two different methods that we can use to evaluate forage quality. The first is a visual assessment. So, we can go look at the hay either prior to purchasing or after purchasing hay. We can go out into our pastures and we can look at, the different forages that are growing and kind of visually assess our forages. Or we can also do an actual forage test or a forage analysis. This can be done both on pastured forages as well as, hays and different harvested forages. And that involves, actually testing those forages for, their different chemical components and getting that nutritional analysis. So that half of the spectrum is what we're going to kind of focus on today, is the forage testing and the analysis side of things. So why is a forage analysis beneficial? Well, there's a few different reasons, and this might depend on, the type of livestock species that you have and kind of your goals for your system. But one potential benefit is, if you're, either purchasing or selling hay, it allows you to assign kind of a market value to the forage. It allows you to determine kind of the worth of that forage nutritionally and make, decisions on, pricing and things like that when you're, buying or selling hay. When it comes to kind of the animal side of things, having that forage analysis allows you to make some of those feeding decisions. You can better, match the quality of the forage to the animal's requirements. We'll talk about that a little bit later in the presentation. And you can also use those, nutritive analysis to, formulate your ration, determine, appropriate supplementation, what nutrients maybe are missing from the forage that you have to supplement in other forms. So it can help make some of those feeding decisions. And then thirdly, one that, you might not consider is something like making storage decisions. If you have a large, purchase of hay and you only have indoor storage for say half of that, you should try to store, the higher quality forage inside to preserve it more, and let the lower quality forage be the one that is outside. So all of these different things can lead to kind of why we might want to do a forage analysis and why we might find that beneficial. So I always like to ask, people, I apologize that this picture is a little bit blurry, but, looking at this picture, which of these hays would you buy? If you want to, go ahead and, type an answer in the chat box. Feel free. But I think we can all agree, looking at these two different lots of hay, that most people would probably choose lot one on the left, right? Visually, it's a little bit more appealing. We would like to think, the nice bright green color means that it has, higher forage quality for our animals. But if we actually look at the forage analysis for these two different hay lots, we can see that they're really similar in their forage quality. That yellow color can be really deceiving. So while those, visual characteristics are important and we can tell some information from them, it's also really important to rely on that actual forage analysis and that forage test to determine the nutrient values. Just to kind of, further iterate that, we forage nutritive value or those actual nutrients present really can't be determined from feel, texture, smell, or appearance. It can only be evaluated with a forage analysis, which again, I like to think of like that nutritional facts label that we see in a lot of our human foods. And just to kind of further stress that point a little bit, a few years ago, there was a group of researchers who had a number of hay samples at one of the big, forage conferences. So they had 4 different hay samples with varying forage quality. So you can see, hay 1, 2, 3, and 4 from lowest to highest in forage quality. And they had, forage industry experts and, forage specialists and, people who've seen a lot of hay samples and looked at a lot of forage samples, look at those different hay types visually and try to guess, which hay was the best just based on the visual characteristics alone. And these percentages show the number of people who ranked that hay type as number 1. So 45% of people thought that hay 1 was the best, when actually it was the worst nutritionally of those 4 hay types. So just to reiterate that point that, the forage analysis is important and it does tell us some things that we can't otherwise tell from a visual appraisal. So one other thing to consider when we're doing our forage analysis and our forage testing is that, you may buy, a whole, wagon full of hay bales, a whole lot of hay. You may be getting hay from, different places or different cuttings. But when it comes down to it, actually how you take those forage samples is really important to consider, because all of that hay that you may see, on that wagon or out in that hay field, when we do the forage analysis, is actually all represented by that, tiny little sample that actually gets scanned, in the lab. So we have hundreds of pounds of, plant material that's really variable, and it all must be represented in a single sample that the lab scans. So, it's important that we're using a proper sampling technique. So we're going to talk a little bit about kind of sampling technique and some do's and don'ts when you're actually going and taking a forage sample. So the first thing, we'll focus on hay first, and then we'll, talk about pasture after that. When it comes to taking a forage sample on hay, one of the most important things is to use a core sample. There are many different types of hay probes. There's, the manual crank ones, there's ones you attach to the electric drill. All of these different types can work. But the important thing is that you're using a core sampler, and ideally it has, that nice sharp tip with, some sort of serrated edge that can cut through that hay. So you're not, pushing different particles of hay out of the way, but you're actually just slicing through that hay and taking a nice core right from the center of that hay. So what you shouldn't do is, take a grab sample. A lot of people like to think, oh, I can just go and grab, a handful of hay from several different bales, and that will be, a good representative sample. But actually, research has shown that those grab samples are usually lower in quality than a correctly sampled forage using a hay core. So if you look at this, chart, you can see that if we use a grab sample instead of a hay core, we're, decreasing we're having a lower value than actually is the crude protein value. So, in this example, it was 19% lower with a grab sample than a hay core. The fiber values are going to be overrepresented, a little bit higher from the grab sample than from the actual hay core. And the energy values are going to be underrepresented. So, and the reason this is a lot of times when we're taking that grab sample, we're maybe losing some of that leaf material, or we may be grabbing more, stems than otherwise. So oftentimes if we are doing a grab sample, it might be a little bit lower in quality than what we actually have present in our hay. So back to the do's, when you're using the hay core, what you should do is insert that probe fully and perpendicular to the stems. So what that means is, for our round bales, we want to sample from the rounded side, not from, the end, the butt end of the hay bale. We want to go perpendicular to those stems. And if we're looking at a square bale, that will be from, the butt end. So we want to go across section, across where those stems are going into that hay bale. We also want to take, a lot of different cores from random bales. We usually suggest a minimum of 20. It can be obviously a little bit more tedious. But the reason we do this is because there is a lot of variation between different hay bales, depending on different, sites of the field, different, how the machinery was working, how the weather was, maybe drying the samples a little bit differently, and just general variability. So, they've done a study like, the graph that you see in the bottom left there was one uniform stack or pile of hay bales, and that was the energy concentration from 20 different samples. And you can see the variation in that energy between each of those individual core samples. So we don't, want to sample, for example, sample number 9 was a lot lower in energy. So that's going to be underrepresented of what we actually have in that pile, versus sample 13 was on the higher end. That's going to be overrepresented. So we want to take many samples from random bales throughout our stack or wagon or whatever, sort of pile that you have, and combine those together. And then, the final sample should be somewhere, usually most labs would like around a half a pound. So if you have too much, when you take all those different cores, then you can mix that all together really well and take a sub sample so you have kind of the right amount to send to the lab. Another thing to consider is how you're handling that sample. So we suggest, collecting it in a labeled plastic bag. It's important to label it so you know where that sample came from, so you don't forget or get samples mixed up. It's best if we store them in kind of a cooler location and not expose them to like heat or direct sun. And then send to the lab, relatively quickly. Hay is a little bit less essential to have a quick delivery than fresh pasture because it's a lot more stable already. But ideally, we send it to the lab, sooner rather than later. So switching gears a little bit to, taking a forage sample from pasture. Similar to hay, we also want to take this from multiple random spots. Again, pastures, just like, hay fields and just like hay bales, they're very diverse. There's a lot of variability, so you may have a pasture with, some areas that are, more leafy, more vegetative, some areas that are a little bit more mature, some areas that maybe were a little overgrazed or undergrazed. So you kind of want to account for all of that variability in your sample. And to do this, you're going to kind of walk through that field in kind of a random or zigzag pattern and pick random spots to sample kind of throughout that field. For the pasture sample, I like to use kind of forage clippers, or they're actually also used as sheep shears, like what you see in the picture there. But really any type of shears or scissors, garden shears that will cut through that forage will work just fine. What you want to do is clip that forage down to kind of that 3 to 4 inch height. That's kind of the recommendation for when we're grazing in pastures. We want those animals to not really graze below that 3 to 4 inches. So we want our forage sample to be relative to what the animals are eating. So we've kind of followed that same guideline for taking our forage samples. And then, all of those different spots that you're sampling throughout the field, what we're going to do is kind of combine all of those forages from each of the spots, mix them together. And then again, if you have too much forage, take a subsample so you get kind of around that half-pound-ish kind of range to send to the lab. For our pasture samples, again, a lot of the same principles apply for handling our samples. So we want to collect these in kind of that labeled plastic bag. We want to keep the collected material cool and out of the sun. And then for pasture samples, I usually suggest people freeze them before sending to the lab. So you're not risking kind of those samples kind of fermenting in that Ziploc bag, or, sitting in the sun and kind of cooking a little bit. So if you're not able to take them, directly to the lab, I usually suggest freezing them and then sending them to the lab that way. As far as, where to send these samples, first off, this is kind of your reminder to make sure that those samples are labeled appropriately. So you need to know, if you have multiple different types of hay that you're sampling, you need to know which type is which sample. If you have different pastures that you're sampling, again, you need to know, which pasture is which sample. The type of forage is important. Most labs will ask, is this a pasture? Is this a hay? Is this a mixed field? Is it a grass only? Is it an alfalfa only? So it's good to know that information, and then the date that you sampled it so you can remember that. And then what you're going to do is send this to an accredited lab. So the National Forage Testing Association has a list on their website of all the different labs that are accredited through their accreditation program. So that's a really helpful place to go and look for a list of forage labs. You can find one that's local to your area. For anyone who is kind of in the Mid-Atlantic region, I also have a list of some of the regional labs on the University of Maryland Extension website. So I have the link there if people are interested in looking at that. All of those labs also appear, on the NFTA website, but it's just a little bit shorter of a list if you're looking for something a little bit more locally. And then whichever lab you choose, all of the labs have some form of submission sheet. So you'll have to go on their website and print off the submission sheet, and that just tells them, who to send the results to, again, what forage type it was, when it was sampled, if it's, hay or pasture, and then what type of analysis that you're interested in. So don't forget to do that and include that with your sample when you're sending it to the lab. When it comes to the type of analysis, there are kind of two main types of forage analysis. So the first is kind of the traditional or more kind of complex, I guess, method, and it's referred to as wet chemistry. It's basically a series of chemical methods that are done on the sample that involve, boiling the sample in various solutions and, drying it and burning the sample to find the inorganic material content, all those different kind of lab methods. And this is really kind of the gold standard of forage analysis. But because you have, all of these different analyses going on, it's a lot more time consuming and it's a lot more expensive. And your sample can really only be analyzed or used one time because, after that sample has gone through the process, you can't kind of backtrack and go backwards. So what the other option is something called near infrared reflectance spectroscopy, or NIRS. NIR, you may hear that term tossed around. So that is basically scanning a sample using light reflectance. And the lab can do this because they have done enough samples with wet chemistry that they've made an equation and it's been calibrated with their equipment. So they can simply scan, the sample and it'll use that calibration equation to actually calculate the nutritive value of the sample. And the benefits of this is it's a lot quicker, it's much more inexpensive, and the process is very quick and easy. The sample can be reanalyzed because it's only being scanned with light. It's not, being soaked and burned and, all those other things. But the downside is it's a little bit less accurate in terms of minerals. It's much more accurate for things like crude protein or, fiber values, but it's not quite as accurate if you're looking for kind of specific mineral concentrations. So those are kind of the two main options, and labs will have other options, different packages that include different things. So it's kind of up to you to decide, how simple or complex you want to go. But usually I suggest if you're just starting out to go with kind of the basic package, and that will give you kind of the main nutrients to start with. So when it comes to the actual forage analysis, here is just an example of one of the samples from one of our pastures here at the research station. You can see when you get the analysis back, there's a whole bunch of different things, like all of these things that you see kind of popping up on your screen now. And I realize it can be, overwhelming, especially if this is kind of your first venture into taking a forage sample. So what we're going to do is go through a few of these. We don't have time to go through all of them, but we're going to go through a few of the main ones that you'll see on a forage analysis and talk about kind of which ones you should focus on first if you're just kind of getting started. So again, I like to think about this as kind of that nutrition facts label that you see on, a lot of our human food at the grocery store. So we'll talk about how these different nutrients for the lab correspond to values on that nutrition facts label. So the first and foremost is dry matter. This is, not really a nutrient, but it's actually the percent of the forage that is not water. So it's the opposite of water. So we know that, pasture or fresh grass contains a lot higher percent of moisture or a lower percent of dry matter compared to hay, because that hay has been cut and dried. So a lot of that moisture has evaporated and gone out of the forage. And as a result, hay has a lot higher percent dry matter compared to pasture. And this is, we'll talk about kind of why this is important, but you'll see on your analysis that you'll have a line showing, the moisture and the dry matter concentration. And you'll see if you add those two up, you'll get 100%. And the key is when you're looking at your forage analysis to focus on that percent dry matter column. So a lot of our, forage nutrients, we always look at them on a dry matter basis. And this is so we can make kind of more equal comparisons. So to just give you an example, if you think about grapes and raisins, grapes obviously have a lot higher percent moisture and the raisins have a lot lower percent moisture. They've been dried, so they have more dry matter. So you can think of kind of the grapes as like the as fed, or the kind of, how the sample is, and the raisin as more of the dry matter. And the reason this is important is because if you have a half cup of grapes and a half cup of raisins, that water adds a lot of volume to what's going into your cup. So as a result, your calories are lower for the grapes because you have more water, versus for the raisins, you have less water, so it's more concentrated and you have higher calories, or a higher percent dry matter. So that's just to give you a little bit of a visual and to remind you to look at the forage sample analysis and focus on that dry matter, on things as a percent dry matter. And again, this just allows us to compare the nutrient content of different feeds on an equal basis. So, in this example, if we were looking at a grass pasture, which is the top row, versus a grass hay, which is the bottom row, if we look at it on that as fed basis, which is including the water component, we might look at these two feeds and be like, wow, these are really different. I should definitely, be feeding this grass hay because it has way higher protein. The pasture is really low. But if we actually compare those two forages on an equal basis or on a dry matter basis, you'll see that they're actually exactly the same. So that water can, influence the concentration of those nutrients. So again, just look at that dry matter column when you're looking at these different nutrients. So getting into the actual nutrients, I've kind of divided this into two different components. The first is kind of the structural components, which is what you'll find kind of in the plant cell wall. And we'll talk about, what those different components are. But the thing to remember is those structural components are more fibrous and less digestible, and they provide the strength and support that that plant needs to grow and stay upright. And then on the flip side, we have more soluble components, which are kind of more of the things that we find inside those plant cells. And we'll go through what some of these are, but these are going to be a little bit more easily digestible, more available nutrients to the animal. So if we first focus on those structural ones, or what is in the plant cell wall, the first one that we're going to talk about is neutral detergent fiber, or NDF. And NDF is basically the cellulose plus hemicellulose plus lignin. So it's kind of all of the fiber components combined together, or all of those primary cell wall components. So the more NDF a forage sample has, the higher the fiber concentration of that forage is. So you can think of that, in terms of the human nutrition labels, like the dietary fiber percent. The higher the dietary fiber, the more fiber is in that product. Something like celery has a lot higher dietary fiber compared to, a French fry. So the higher the NDF, the higher the fiber content. And this is important because it affects the intake of that animal. So the animal is not able to consume as much. If there's a lot of fiber, it's, filling them up. They have a lot of gut fill. They're going to have lower intake. So those have an inverse relationship. The next one is acid detergent fiber, which is, also, again, one of the fiber components. But this is just the cellulose and lignin. And the reason we kind of set it up this way is those are the least digestible of all the different types of fiber. So this measure is looking at not just all the fiber, but which of those fibers are the least digestible, or the least, able to be broken down in the GI tract of that animal. So again, it is a positive relationship with ADF and fiber, because it is one of our fiber values. But instead of relating it more with intake, ADF is usually related more with digestibility, because it's those less digestible portion of that fiber. So switching from kind of more of those two structural components to more of kind of the interior of the plant cell, one of the first ones people like to look at is the crude protein. So we know that protein provides some of the essential amino acids. There are, for different species, there are different ones that are more limiting than others. But crude protein as a whole is actually not directly measured. There's no lab analysis that actually, directly measures the crude protein of the forage. It's actually estimated based on the nitrogen content, because all protein, all amino acids contain nitrogen. So they use that nitrogen to estimate the total amount of crude protein. And this includes, as a result, this includes both true protein, so actual crude protein, as well as a little bit of non-protein nitrogen. We can have, other sources of nitrogen in that plant, but because we're just measuring overall nitrogen, keep in mind that that's why it's called, crude protein. It's not a perfect measure, but it's, the estimate of the protein. So again, tying that back to kind of the human nutrition label, this one is kind of an obvious comparison. Crude protein is like the protein that we see on our human nutrition labels. Another one of our kind of interior cell components is the non-structural carbohydrates, or NSC. And this is a measure of kind of more of the simple carbohydrates like the starches and the sugars. These are basically a readily available energy source. So they are rapidly and easily digested. So, there you have kind of the sugars and the starches that are inside. If you don't see the non-structural carbohydrates, you may see some of the other carbohydrate components, and you can add those together to get your non-structural carbohydrates. So for the comparison to kind of humans, this would be like your total sugars, the sugars that you see on that label. So next we're going to move into kind of measures of energy. So one of the main measures for energy that we see for forages or feed is total digestible nutrients, or TDN. And this basically means the energy that's available after digestion. So how much energy was that animal able to extract from that forage? Kind of like crude protein, it's not something that we can measure directly, but it's calculated based on a whole bunch of other components like protein, fat, carbohydrates, digestible fiber. All those are kind of added together to get the total digestible nutrients. So there is an equation for this, and it does differ a little bit based on the type of forage. So there's, a different equation for legumes than there is for grasses. But just remember that it's kind of the overall energy value of that feed. So for, the human comparison, it would be kind of like the calories, the total calories that you're able to get from that particular feed. Another measure of energy is using the net energy system. So there are a whole bunch of different, categories in this, but this is a different way to look at the energy, and it's accounting for different loss pathways. So, we have, the potential energy or the gross energy of a feed. And then if we take away, energy that's lost in the feces, because we know some energy, leaves the animal that way, then we have digestible energy. If we take away energy that's lost from gases or urinary output, then we have metabolizable energy. If we take away the energy that the animal is using for heat, to maintain their body heat, then we kind of have a net energy. And then if you really get specific, you can break this down into the net energy that those animals need for maintenance or for production, whether that's growth or milk production. So really this pathway kind of separates the energy requirements into those different components. It's a different way of looking at energy that kind of breaks it down a little bit further based on those different components. And depending on what species you have or what species you work with, you may use TDN or you may use one of these energy components. So for example, the cattle and the swine industry are, further down the spectrum. They use net energy oftentimes for their ration balancing. But in the equine world, we're still kind of back up on the digestible energy. So all of these are different measures of energy, kind of just depends how specific you get with it. So again, just, to kind of summarize, this is a little bit more of a visual way of looking at it. The digestible energy is the energy available for use after those fecal losses. It's more common in the equine or in poultry. And you can kind of, picture it as, the gross energy is everything, the potential of that feed that goes into the animal. The digestible energy is what is absorbed, and then minus the output, or what goes out in the feces. All right, so the kind of final quality term that we're going to talk about is relative forage quality, or RFQ. And the reason I want to bring this up is, we kind of talked in the beginning how quality means more than just what are the nutrients present in that forage, right? A high quality forage does need to be something that's highly digestible or have, a good high energy concentration, but it also needs to be something that is readily consumed. So we have a high intake. So the goal with relative forage quality was to come up with a single measure to represent the overall quality, which would allow you to kind of more easily compare across different forages, across different types of forage, all those different comparisons. So this is another measure that's really not directly measured, but is calculated based on some of the different values. And it accounts for the TDN, or the total digestible nutrients, and also the dry matter intake. And there you can see, what the equation is. There's a lot of different components that go into it, but just think of it as kind of that single measure to represent forage quality. So to again visualize that a little bit better, if we have, two piles of hay that are the same weight, on the left we have a pile of alfalfa, on the right we have a pile of Bermuda grass. You can see that the energy content based on the weight is very similar. We have a TDN right around 60 for both of these piles of hay. So for relative forage quality, what's the difference here? Well, we have a similar TDN, so we have a similar energy content, but if you look at the pile volume, it's a lot bigger for the Bermuda grass. The RFQ, as a result, is higher for the alfalfa than for the Bermuda grass hay. And why is this? Well, the alfalfa pile has a higher RFQ because they can eat more of it. You're able to consume more of the alfalfa, so they're able to consume more total energy, as a whole than they are from the Bermuda grass. So again, to try to kind of simplify that a little bit, or relate it to kind of something that, we might think about as humans. If you think about this as like a plate of French fries versus a plate of peppers, you can put an equal amount of energy on both of these plates. But the amount of French fries you have to eat to get that energy or those calories is a lot smaller than the amount of peppers you would have to consume to get the same amount of energy or the same amount of calories. And I don't know about you all, but I would much more easily be able to consume this small plate of French fries than I would that whole plate of peppers. So it just is a way to kind of combine all of these factors into one term and use that to compare our different forages. So to kind of summarize, I know we just went over a lot of different nutrients that are at play with our forage test. But I wanted to pick out just a few of the key ones that I would start with if I were kind of just getting into this and just kind of looking at my first kind of forage sample. The first is fiber. So look at that neutral detergent fiber value. I realized that it's hard to say exactly what percent NDF is good and what percent NDF is bad, because it depends on what type of forage it is. It depends on the animal that we're feeding. It depends on a whole host of factors. But just to give you a really rough range or a place to start, we want something that's not too fibrous, so we're able to have those animals consuming enough that they are able to eat, and not get too full or have too much gut fill from too much fiber. So I would say in general, for like a grass hay, an NDF that's less than 50% would be on the lower end, and NDF that's greater than 60% would be on the higher end. And this scale will be shifted down a little bit for legumes because they have a little bit less NDF as a whole than a grass would. The next one is energy. So again, depending on which species you have, there are different measures that you might want to focus on, whether that's TDN or digestible energy or net energy. If we look at just TDN for this example, we want something that contains enough energy that we're providing the calories that those animals need for maintenance or growth or whatever their life stage is. So on average, we could say maybe that a TDN of less than 55 is on the lower end of the energy spectrum, and a TDN of greater than 65 is on the higher end of the energy spectrum. And then for protein, also, of course, we want to make sure we're providing enough of those essential amino acids and enough, protein as a whole for that animal. So I would say something less than 10% would be on the lower end, and something higher than 14% would be on the higher end. So that just gives you kind of some rough ranges as a place to start. And then RFQ, of course, is not really an essential nutrient. There's not a specific amount of RFQ that we need, but it's really useful for comparisons. So if you're looking at two different forage types, you can look at the RFQ and see, which one has a higher overall quality value between those different forages. So, all that said, I've mentioned several times how forage quality is variable, whether that's hay or pasture or anything. And this is just a whole list of factors that can contribute to that variability in that forage quality. And some of these things we can, visually see, the stage of maturity. If you're looking at a pasture, you can see, are they higher in maturity or lower? Some of the species differences. And some of them come in more on the production side. So, if this is hay we're talking about, it would be like the hay producer. How is the soil fertility in that field? How was that hay harvested under good conditions? Are there any, anti-nutritive factors to consider? All of these different things can affect the overall quality of the forage. So I just really briefly wanted to go through a couple of the main ones to give you just some idea of kind of some things to look for. If we start with forage maturity, just remember, if nothing else, that this is kind of the biggest determinant of nutritional value. So maturity plays a huge role in the nutritional value of our forages. Forages that are more vegetative, more leafy, kind of greener, less stemmy, are higher in energy and higher in protein and higher in, some of the vitamins and minerals. And forages that are more mature or more stemmy will be, lower in energy and higher in those fiber values. And this plays a role in, not only the intake of that forage, but also how much of that is able to be digested. So here we have different growth stages. So at the top of the list was a more vegetative, less mature, and as we move down was a more mature, more stemmy, more fibrous forage. And you can see that, intake was higher when those forages were more vegetative and lower as we got, to a more mature forage. And digestibility was also higher when the forages were more vegetative and more leafy, and lower when those forages got more mature. So higher maturity is lower intake and lower digestibility. So remember that plays a really big role. Another big one is the leafiness, or you may hear of like the leaf to stem ratio. So we know that leaves are like the most nutrient rich part of the plant. They're higher in quality. There's more energy and protein in the leaves. There's less fiber in the leaves. And the leafiness is also closely tied to maturity. So as our forages mature, as they move to the right across this graph and they're increasing in maturity, you can see that the leaf percentage is decreasing and the stem percentage is increasing. And, leaves can be lost during the haymaking process, depending on how hay was made. This also plays a role in, pasture. The same thing happens as pasture plants are growing and maturing. And there is, in general, if you're looking at hay, you would want to see more leaves compared to stems as an indicator visually that hay is a little bit higher in quality. Another big one is differences in forage species. So depending where you are and what's, growing in your area, you may have cool season forages, warm season forages, grasses, legumes, perennial plants, annual plants. There's a whole host of different forage species that are grown and fed to livestock. And one of the biggest things to remember is that there are kind of some rough differences between these different types of forages. So if we look here, at a lot of those nutrients that we had just talked about, we can see that legumes tend to be a little bit higher in energy compared to our grass species. They also tend to be a little bit higher in protein. And a warm season grass tends to be a little bit lower in protein. The fiber values tend to be highest for our warm season grasses and lowest for our legume species. Things like calcium tend to be higher in our legume species, so they have a little bit more minerals like calcium. And the non-structural carbohydrates, or those, sugars and starches, tend to be a little bit lower in warm season grasses and legumes compared to our cool season grasses. So for overall quality, just in general, legumes tend to be the highest, warm season grasses tend to be the lowest, and cool season grasses tend to fall kind of somewhere in the middle of that spectrum. But again, this is, relative to all of those other factors, stage of maturity, how the forages were handled, all those different things play a role. Just to show you that, there is a lot of variability there. This is some samples that were submitted to the University of Georgia Forage Lab. And you can see the different types listed along the left-hand column. And the dark green bar kind of in the middle of each of the samples is the average, but the range of color, going from left to right, from yellow to green, is the variation. So you can see that even within a small category like our small grains, there's a lot of variation in the quality based on those different samples. But in general, if we look for those same kind of trends, here's our kind of cool season perennials, fescue and orchard grass. Here's our warm season perennials, which tend to be a little bit lower. And then here is more of our legumes, which tend to be a little bit higher. So you can still pull out those same kind of general trends, but just know that there is a lot of variation going on. And then finally, this is kind of for, people who are more on the production side, if it's hay or if you have pastures. Things like fertility can play a role in the quality. We know that, soil pH can affect the availability of our nutrients, and fertilization with nitrogen will not only affect the yield of our forage, but also the crude protein levels in that forage. So fertility can play a role in the overall forage quality. And the harvest and storage techniques. So, how is that hay made? How is that hay dried? How was it stored? Was it stored properly, or did it have a lot of weathering? All of those different things can also play a role in that end result going into that forage quality sample. So, here's our whole list again. It's a lot to think about. There's a lot of factors that go into it. So a lot of people, ask, okay, well, all of that said, okay, there are all these factors, but what do I do? I have these 2 different forage tests here. How do I know which one should I feed? Which hay should I buy? What should I purchase? Well, again, that, it depends on a lot of different things, right? First and foremost, we know that the nutritional needs vary by animal. So, different animals have different body conditions, different ages, different breeds, different activity levels, and that will all play a role in their nutrient requirements for that animal. But if we look at kind of the general trend, here we have a scale, moving from left to right of increasing quality that those types of livestock need. So say, at the low end are things like a dry cow or an idle horse that don't need quite as much quality or nutrients in their diet as something like a lactating beef cow or a growing heifer. Those animals have a little bit higher nutritional needs. And then, at the highest end of the spectrum, we have, a lactating dairy cow or a very young, calf that's just starting growing. Those animals will have the highest nutritional needs. So what we can do is kind of try to match those species with our forage quality test, right? So in the left 3 pictures, we have kind of our lowest end of the spectrum. We have, that horse that's idle. We have the mature beef cow that's, just out in the field, in between pregnancies maybe. We have maybe a mature bull who's not breeding right now. He's just, waiting for the next breeding season. So those animals are more on the maintenance end, and they'll need, a higher fiber forage, something with lower energy, lower protein. They're just kind of maintaining. In the middle, we have maybe a horse that's under moderate work, or we have, an animal that's growing, but it's not quite as young anymore. So it's kind of at the end of that growth spectrum. Or we have an animal that's pregnant, but not lactating yet. So kind of those moderate stages maybe need something with more moderate fiber, moderate energy, and moderate protein levels. And then, of course, at the highest end of the spectrum, we have things like, animals that are lactating, especially the ones that are in the peak of their lactation cycle, and we have young growing animals, and we have, you know, those really high demand athletic performance horses. Those animals are going to need something with the lowest fiber, the highest energy, and the highest protein that we can give them. So, you know, it varies. Size and body weight can also play a role. You know, a smaller size will need less nutrients than a larger sized animal. Environment can play a role. You know, cold winter weather, those animals will need a little bit more calories to be able to maintain their body heat than an animal in the summertime. And body condition score, you know, an underweight animal will need a little bit more calories to bump up that weight than an animal that's overweight and maybe needs a little bit less calories so they lose a little bit of weight. So all of those different things play a role, but really what it comes down to is evaluating it and making adjustments. So look at the body condition score of your animals, look at their weight and how it's changing over time, look at their performance. You know, there are a lot of different things that we can measure, you know, weight gain, reproduction traits, production, whether that's milk or growth, athletic ability. You know, do some measurements, track some of those things over time, look at health or disease incidents and feed intake, and make adjustments as needed. You know, it's always going to be a little bit of a moving spectrum when it comes to forages and forage quality. So I know that was a lot. Hopefully I didn't lose anybody in there, but basically to kind of summarize, we know that our forage testing allows for an accurate quality assessment. So it's really important that we're doing that. When we're thinking about forage quality, if we're just starting out, we want to focus on maybe some of the key things like NDF for the fiber, TDN for the energy, and crude protein. And then we can use those helpful values like RFQ to make our comparisons across different forage species or forage types. Remember that there is a lot of different factors that influence this, you know, maturity, leafiness, forage species, all of those will play a role in that end forage quality. And what it comes down to is selecting an appropriate forage to match our animals' needs. So looking at those different values for fiber, energy, and protein, picking a lower quality forage for animals that have a lower nutrient demand and a higher quality forage for animals that have a higher nutrient demand. And then again, evaluating and adjusting as needed. So with that, that was everything that I had for today. I did put my contact information up here in case anybody has any questions. You know, if they think of later, feel free to, you know, email me or give me a call if you have questions, and I'm happy to take any questions now if there are any.
That was wonderful, Amanda. Thank you very much. Give people a little bit of time if they have questions for Amanda. I think I really like the insight about looking at visually versus having a forage analysis. I think that's a really good example.
Yeah, there's definitely things you can tell, you know, visually, but really you can never tell the nutrient concentration, you know, just from looking at it. So it's always important to use both, use all of your tools in your toolbox, if you will.
Yeah. All right, I've got one here. Do you have any recommendations for summer cover crop species for renovating a slope in a horse pasture?
Okay, yeah, read that one more time. Summer cover crop species.
So, yep, summer cover crop species for renovating a slope in a horse pasture.
Okay. So yeah, horses are, there's a lot of interest in using annual forages for livestock, whether that's, you know, during a renovation process, or some people are just, you know, setting aside a few acres or, you know, portion of their pastures to plant annual forages, because you can do things like plant a summer annual that will produce a lot during the summer when some of our cool season forages are going through kind of that, you know, summer slump. Horses are a little bit more particular, I guess I will say, than other livestock because they have a little bit of other nutritional concerns. So due to those, there's a few species that we kind of focus in on for horses. One is the pearl millet. That can be a good summer annual that can work well for horses. They do also make dwarf varieties of it that grow a little bit lower and are better kind of for grazing animals. They don't get quite as tall. And then another one actually that some people may cringe when I say this, but they have developed some new varieties of crabgrass that are not kind of as weedy or invasive as the wild type one, but are actually forage varieties of crabgrass. So they're meant to be a little bit more leafy, more productive, and people are actually planting those and finding them as good forage options for producing some of that summertime forage. And the advantage to crabgrass is it's more similar to our other kind of traditional perennial species, just in its growth habit. So horses may find that a little bit more appealing than something that's a little bit outside of their comfort zone, if you will. Those would be my kind of two best guesses. The third one would be a teff grass. That one can be a good forage for horses, and again, it's kind of finer and leafier and good quality, makes a good summer feed. The only caution with that one is it can be a little bit harder to get established. So people can have trouble. It's a really small seed, it's very fine, so it can be a little bit trickier to get a stand established with that.
Great. That was a great explanation. And there are so many different species. I mean, especially when you start broadening your horizons outside of maybe some of the traditional cool season grasses. So that's excellent.
Yeah, maybe that will have to be a topic for next year's forage
Oh,
webinar.
yeah. Yes, that would be
Annual
great.
forage options.
Yes. Great. Well, I don't see any other questions. Some great comments about helpfulness of this webinar, and you've got her Maryland forages, and there's some great information available on that's shared on the Facebook page. So definitely follow that. Oh, I do see another question. Okay. So yes, this is being recorded, and we will be sharing it out. Anyone that registered for today will get the set of slides as well as the recording from the video, so you can listen to it later. We have a Mid-Atlantic Women in Ag YouTube channel, so you can subscribe to that too, and you can see when we post new videos. But yes, we'll be sending out to your email through the Eventbrite distribution.